Before going back to the e-text, there is another reading for Module 2.3, which is functional specialisation. Zeki put forward a functional specialisation theory. According to this theory, different cortical areas are specialised for different visual functions. The visual system resembles a team of workers, each working on his or her own to solve part of the complex problem. The results of the labours are then combined to produce the solution, i.e., coherent visual perception. Why might there be functional specialisation in the visual brain? Zeki suggested two reasons. First, object attributes occur in unpredictable combinations. For example, a green object may be a car, a sheet of paper or a leaf, and a car may be red, black or green. Thus, we must often process all an object's attributes to perceive it accurately. Second, the required processing differs considerably across attributes. For example, motion processing involves integrating information from two or more points in time. In contrast, form or shape processing involves considering the spatial relationship of elements to each other at one point in time. The organisation of the main visual areas in the macaque monkey is shown in figure 2.7, which is on the next page, page 43. The organisation of the human visual system closely resembles that of the macaque, and so reference is often made to human V1, V2, and so on. Here are the main functions Zeki ascribed to these areas. V1 and V2. They are involved at an early stage of visual processing. They contain different groups of cells responsive to colour and form. V3 and V3A. Cells in these areas are responsive to form, especially the shapes of objects in motion, but not colour. V4. The majority of cells in this area are responsive to colour. Many are also responsive to line orientation. V5. This area is specialised for visual motion. In studies with macaque monkeys, Zeki found all the cells in this area were responsive to motion but not colour. In humans, the areas specialised for visual motion are referred to as MT and MST. Zeki assumed that colour, form and motion are processed in anatomically separate parts of the visual cortex. Much of the original evidence came from studies on monkeys. Relevant human evidence is considered below. Form processing. Several areas are involved in form processing in humans, including areas V1, V2, V3 and V4, and culminating in inferotemporal cortex. There is evidence that neurons in inferotemporal cortex respond to specific semantic categories, example, animals, body parts. See Chapter 3. There is also strong evidence that neurons in inferotemporal cortex are involved in form processing. In one study, neurons within inferotemporal cortex responded to three-dimensional object shape. Baldassi and others measured neuronal activity within anterior inferotemporal cortex in two monkeys. Many neurons responded on the basis of aspects of form or shape: round, star-like, horizontal, thin, pointy, vertical, thin, rather than object category. If form processing occurs in different brain areas from colour and motion processing, we might anticipate some patients would have severely impaired form processing but intact colour and motion processing. That does not seem to be the case. According to Zeki, a lesion large enough to destroy areas V3, V4 and inferotemporal cortex would probably destroy area V1 as well. As a result, the patient would suffer from total blindness rather than simply loss of form perception. There's two key terms here. Achromatopsia, and that's a condition involving brain damage in which there is little or no colour perception, but form and motion perception are relatively intact. And achinotopsia, a brain-damaged condition in which motion perception is severely impaired, even though stationary objects are perceived reasonably well. Colour processing. The assumption that V4 is specialised for colour processing has been tested in several ways. These include studying brain-damaged patients, using brain imaging techniques, and using transcranial magnetic stimulation, TMS, to produce a temporary lesion. We will discuss these three kinds of studies in turn. Suppose area V4 and related areas are specialised for colour processing. If so, patients with damage mostly limited to those areas should show little or no colour perception with fairly intact form and motion perception and ability to see fine detail. This is approximately the case in some patients with achromatopsia, which is one of the key terms I mentioned before. Bouvier and Engel carried out a meta-analysis involving all known cases of achromatopsia. A small brain area within ventral, bottom, occipital cortex in or close to area V4 was damaged in nearly all these cases. The loss of colour vision in these patients was often only partial, suggesting V4 is not the only area involved in colour processing. In addition, most patients had significant impairments of spatial vision. Functional neuroimaging evidence that V4 plays an important role in colour processing was reported by Goddard and others. Observers viewed movie segments presented in full or no colour. There was substantially more activation in ventral V4 with the full colour segments. Wade and others had previously found that V4 was actively involved in colour processing, but other areas, V1 and V2, were also activated. Banassi and others found that performing a task on a stimulus, diamond shape, was faster when preceded by a prime in the same colour. However, this priming effect was no longer present when transcranial magnetic stimulation, TMS, was administered to V4. This finding occurred because TMS reduced colour processing in V4. In sum, area V4 and adjacent areas are undoubtedly involved in colour processing. However, V4 is not a colour centre. First, V4 is a relatively large area involved in spatial processing, shape processing, and depth perception, as well as colour processing. Second, some ability to process colour is present in most individuals with achromatopsia and monkeys with lesions to V4. Third, several areas outside V4, including V1 and V2, are also involved in colour processing. Motion processing. Area V5, also known as Motion Processing Area MT, is heavily involved in motion processing. Functional neuroimaging studies indicate motion processing is associated with activity in V5 or MT, but do not show that V5 or MT is necessary for motion perception. This issue was addressed by McKeefrey and others, who used transcranial magnetic stimulation to disrupt motion perception. When TMS was applied to V5 MT, it produced a subjective slowing of stimulus speed and impaired observers' ability to discriminate between different speeds. Additional evidence that area V5 MT is important in motion processing comes from research on patients with achinotopsia. Achinotopsia is a condition in which stationary objects are perceived fairly normally, but motion perception is grossly deficient. Zill and others studied LM, a woman with achinotopsia, who had suffered bilateral damage to the motion area, V5 MT. She could locate stationary objects by sight, had good colour discrimination, and her binocular vision was normal. However, her motion perception was grossly deficient. She had difficulty in pouring tea or coffee into a cup because the fluid appeared to be frozen like a glacier. In addition, she could not stop pouring at the right time since she was unable to perceive the movement in the cup or a pot when the fluid rose. In a room where more than 2 people were walking, people were suddenly here or there, but I have not seen them moving. They were her words. Cooper and others reported the case of a 61-year-old with achinotopsia. She perceived static objects normally, but smooth movements of people were seen as freeze frames. People close to the opening of a train door appeared to move in slow motion. V5 MT is not the only area involved in motion processing. There is also area MST, medial superior temporal, which is adjacent to and just above V5 MT. Vayner studied 2 patients with damage to MST. Both patients performed normally on some tests of motion perception, but had various problems relating to motion perception. One patient, RR, frequently bumped into people, corners and things in his way, particularly into moving targets, example people walking. These findings suggest MST is involved in the visual guidance of walking. There is an important distinction between first-order and second-order motion. With first-order displays, the moving shape differs in luminance, intensity of reflected light, from its background. For example, the shape might be dark, whereas the background is lit. With second-order displays, there is no difference in luminance between the moving shape and the background. In everyday life, we encounter second-order displays infrequently. Example, movement of grass in a field caused by the wind. There is an ongoing controversy as to whether different mechanisms underlie the perception of first-order and second-order motion. Evidence that different mechanisms are involved was reported by Ashida and others. Repeated presentations of first-order displays led to a substantial reduction in activation in motion areas MT and MST. This adaptation occurred because many of the same neurons were activated by each display. Very similar reduction in activation in the motion areas occurred with repeated presentations of second-order displays. However, there was no evidence of adaptation in MT and MST when first-order displays were followed by second-order displays or vice versa. The implication is the two display types activated different sets of neurons and thus probably involved different processes. Support for the notion of different mechanisms for perception of first-order and second-order motion was also reported by Rizzo and others. They identified 22 patients with a deficit in perception of first-order but not second-order motion, and one patient with a deficit only in perception of second-order motion. This double dissociation suggested different processes are involved in perception of the two types of motion. However, some evidence suggests first-order and second-order motion perception depends on the same mechanisms. Hong and others found direction-selective responses to both types of motion in several brain areas, V1, 2, 3, 3A, 4, and MT+. These findings suggest none of these areas is specialised for processing only one type of motion. More importantly, the patterns of activation associated with perception of first and second order motion was similar. Further support for the same mechanism assumption was reported by Cowley and others. They disrupted activity in V2 and V3 or V5 MT plus via transcranial magnetic stimulation, and found this led to impairments in the perception of first order and second order motion. How can we reconcile the various findings? First and second order motion perception probably depend on similar but not identical underlying mechanisms. That assumption provides a potential explanation for the apparently inconsistent findings in this area. Binding problem, and this is a key term, meaning the issue of integrating different types of information to produce coherent visual perception. Zeke's functional specialization approach poses the obvious problem of how information about an object's motion, colour, and form is combined and integrated to produce coherent perception. This is the famous binding problem, which concerns how items that are encoded by distinct brain circuits can be combined for perception, decision, and action. One approach to the binding problem is to argue there is less functional specialization than Zeke claimed, which would reduce the complexity of the problem. For example, Seymour and others presented observers with red or green dots that rotated clockwise or counterclockwise. Assessment of brain activity indicated colour motion conjunctions were processed in several brain areas, including V1, 2, 3, 3A, B, V4, and V5 MT+. Thus, there is extensive binding of colour and motion information even early in processing. Feldman argued that there are actually several binding problems. There is the problem of how visual features are bound together. Another problem is how we bind together information over successive eye movements to achieve the subjective perception of a stable visual world. Within the above broader context, it is clear that many different lines of research are relevant. For example, observers need to work out which parts of the visual information available at any given time belong to the same object. The Gestaltists put forward several laws to account for how observers do this. Research on selective attention, especially research on visual search, detecting target stimuli among distractors, is also relevant. This research shows the important role of selective attention in combining features close together in time and space. One approach to solving the binding problem is the binding by synchrony hypothesis. According to this hypothesis, detectors responding to features of a single object fire in synchrony, whereas detectors responding to features of separate objects do not. Of relevance, widespread synchronization of neural activity is associated with conscious visual awareness. The synchrony hypothesis is oversimplified. Visual processing of an object occurs in widely distributed areas of the brain and proceeds through several stages. This makes it implausible precise synchrony could be achieved. Another problem is that two or more objects are often presented at the same time. On the synchrony hypothesis, it would seem hard to keep the processing of these objects separate. Gutman and others suggested an alternate hypothesis based on the notion that perception depends on patterns of neural activity over time rather than on precise synchrony. In sum, there are several binding problems, most of which are hard to solve. However, progress has been made. Selective attention undoubtedly plays an important role. Feature binding is typically associated with synchronized activity in different brain areas, but the association is often imprecise. There is also the issue of explaining why and how synchronized activity occurs. Evaluation. Zeke's functional specialization theory has deservedly been influential. It is an ambitious attempt to provide a simple theoretic framework within which to understand a remarkably complex reality. As is discussed later, Zeke's assumption that motion processing typically proceeds somewhat independently of other types of visual processing has received reasonable support. There are three main limitations with Zeke's theoretical approach. First, the brain areas involved in visual processing are less specialized than implied by the theory. Hayward and Cowie considered the percentage of cells in each visual cortical area responding selectively to various stimulus characteristics. See Figure 2.8, which is on page 47. Cells in several areas respond to orientation, disparity, and colour. Specialization was found only with respect to responsiveness to direction of stimulus motion in MT. Second, early visual processing in areas V1 and V2 is more extensive than suggested by Zeke. As we saw earlier, Hegde and Van Essen found many V2 cells responded to complex shapes. Third, Zeke and his colleagues have not yet solved the binding problem. In fairness, there are probably several binding problems and no one has solved them. However, it appears that selective attention is of major importance. Two visual systems, perception and action. There's a few key terms here. There is the ventral stream,